this computer.
Okay. So do you all see my screen?
Yes.
Yes.
Good. Okay. So we are going to learn about. So we are going to learn about genes and development in this module. And as I told you earlier, this module is all about how do you define a gene, how the information is stored within a gene, which is basically a piece of DNA which encodes specific coding information or non-coding as well, as we are in 21st century now. We are going to learn about how this information is decoded from DNA into RNA and eventually into a protein. So this flow of information from DNA to RNA and eventually into a protein is also referred to as central dogma in molecular biology. We are also going to learn about how information stored within DNA is replicated, or how DNA molecule is replicated and passed on to the progeny, whether it's through mitosis or meiosis, etc. So the inheritance, we are also going to learn about inheritance, how genes are inherited, and what effects we may see if there are changes or alterations in the DNA sequences. It can be non-significant all the way to highly significant, which may cause death as well. So in addition to learning how information in DNA is stored and how this information is decoded or replicated, one of the important part of this module is about inheritance, genetic inheritance. How do we study inheritance of traits and characters? How do we use variation, alterations, changes, mutations in DNA to understand function of a gene? Okay, and last but not the least, we are then going to wrap all this module by understanding the genetic basis of development. We are going to then look at the whole organism level to see how information stored in DNA is decoded in a very, very systematic manner, in a very specialized way, which leads to development of cell fates. We develop different kinds of cell types. For example, we have epithelial cells, we have lungs, liver, kidney, heart, eyes, etc. All this develop from single cell, which is fertilized egg, which contains 46 chromosomes in our case. So how the genetic information or information present within the DNA is decoded, that from single cell we eventually get more than 200 different cell types in our body. So as I said, we call this module as genes and development. So we start from simple DNA molecule, the information stored in DNA molecule, how it is replicated, all the way to the genetic basis or molecular basis of development. So we learn all this in this module. So we are going to start today's lecture. Today we are going to learn about DNA molecule, how DNA molecule stores information, and how this information is duplicated or replicated. Um, so we have two words here on the slide, which will come repeatedly in this module in next 5 days, is the genotype and phenotype. So genotype is basically collection of all the genes you have in a single cell or in an organism. And phenotype is the appearance. From genotype to phenotype means, you have, for example, within nucleus, you have in humans, imagine we have these 46 chromosomes. So all the genes which are present on these 46 chromosomes, they are similar among humans, but they have very, very specialized or specific differences, which we refer to as variation as well. So each one of us may have a characteristic genotype, the collection of genes, okay? And collection of all the genes is also referred to as genome, okay? The way we develop, or the way different organisms develop or species develop. For example, here you have strawberries, you have, these lemons, there are different fruits here. Here you can see, so okay, strawberries and lemons are different fruits, these currants are different, berries are different fruits. So one can say, yeah, they contain, different genotypes or genomes, okay? They differ in the DNA sequence, number of genes, number of chromosomes, etc. However, if you look at this one, the reddish, the carrots, what we call, so these, they all are same species. They all have same number of chromosomes because they just differ in the color, let's say. Now, it means there is variation. They all may contain same number of n number of chromosomes, but within their genome or the collection of genes they have, there must be some very specific variation. And that variation is decoded, you can see in the different colors, orange, white, red, more purplish. So the appearance, physical appearance, is referred to as that word is called phenotype. Okay, from genotype to phenotype. So this whole module I told you is about genes and development. So from here we go from genes, and then we are talking about phenotype, which means physical appearance of a fruit like this or this vegetable, or humans. Look, I have much more gray hairs than many of you. Let's say there could be a person who has blue eyes, you may have brown, another one may have black eyes. We all are humans, we all are same species, but we carry variation in our genome as well, which is decoded, and then we develop. That's why I said genes and development. And we develop a specific kind of phenotype, which could be blue eyes, green eyes, brown, etc. We are going to learn a great detail about all these processes. But in order to understand all this, we need to understand first DNA. We need to first learn about, what is DNA, why we say there's a code, why we say there is information hidden or stored in the DNA. So in order to understand all that, we have to understand the basic molecule of life, which is called DNA. So in these two lectures. So today and tomorrow, you are going to learn about DNA or proteins as molecules of hereditary. We will define what hereditary means. Hereditary basically means, humans. So it's basically continuity of the life in a species. It means humans will always have babies which are identical to humans. Dogs will always have pups. Cats will always have kittens. Okay, and you won't have kittens or pups in your family. You will always have babies, human babies. Okay, so this concept of hereditary is basically due to very specialized DNA sequences we all carry. And as I said, DNA sequences are decoded into proteins, which means, so like we have all the collection of genes is called genome. All the collection of proteins in a cell is called proteome. Now, each species has characteristic genome and a corresponding proteome. Now, if we have to learn what molecule is responsible for hereditary, whether it's DNA or protein. 100 years ago, this was a big question. This was a big question whether DNA molecule or proteins, which one of them is molecule of hereditary, which defines inheritance. Okay. Then we will learn about how information is stored in DNA, how DNA is copied, because to grow and to reproduce, it's very important that, individual cell, it divides, and when a cell divides, it means it has to divide its genome as well. Let's say you are a single cell. This cell divide into two cells. So all the DNA which was present here will undergo replication and then will be passed on to daughter cells. So how this hidden information is decoded, and how all this governs inheritance, this is what we are going to learn today. So these are two examples. For example, here you have what kind of cell is this, and what kind of cell is this? Who would like to answer?
Sir, the right one is prokaryotic.
Very good. The right one is
The left
prokaryotic.
and left one is eukaryotic.
आपका क्या नाम है?
Adil Akram.
Adil. Ji Adil, why do you think that left one is prokaryotic and right one is eukaryotic?
Sir, the left one is prokaryotic because its genetic material is just floating inside the membrane, or it doesn't have a defined nucleus wall around its genome. And the left one is eukaryotic because it has defined nucleus wall and it has genetic material packed inside.
Very good. So here we have two kinds of cells. This is a prokaryotic cell, okay? And the reason it is prokaryotic, and this one is eukaryotic cell. So in eukaryotes, you have membrane-bound organelles. For example, this one is nuclear membrane. These ones are membranous organelles like endoplasmic reticulum. If you go deep down, you will see, mitochondria, Golgi apparatus, etc. But in a prokaryotic cell, which is a A simple most example is bacteria, for example. Bacteria is a single cell living organism. So you can see here this cell, this is a bacterium, and there is no membrane bound nuclear structure or any other organelle. And this is what the nuclear genome is. All the DNA is like, in a supercoil, and it's mostly a circular molecule. Bacterial genome or prokaryotic genome is basically a circular molecule, circular genome. In case of eukaryotes, we have, genetic material or DNA is present in the form of chromosomes. This circular molecule in bacteria is also called a chromosome, but it's a one giant single chromosome. But in case of eukaryotes, it depends upon which organism we are talking about, which organism is under study. There can be different number of chromosomes. For example, in humans, we have 46 chromosomes. In case of fruit flies, for example, we have 4 chromosomes. In case of maize plants, we have, I believe, 9 chromosomes. 9? No. A diploid is 18 chromosomes. Okay. And in peas, for example, 7 chromosomes. And the chromosomes normally are present like this. Now, this chromosome, if you rip it apart, if you tear it apart, you can see it's made up of double helix. You can see this is the double helix DNA, okay? Like this. DNA in prokaryote is also exactly similar way. It's a double helix, a circular molecule. However, in case of eukaryotes, DNA is wrapped around specialized proteins which are called histones. Okay. There are 4 specific histones: histone H2A, H2B, H3, and H4. 2 molecules of each. So a tetramer of this and 2 dimers of this, total 8 proteins. They make a circular pulley-like structure around which this DNA, you can see, is being wrapped around. It's going like this, twice, like this. This is his nucleus. This is a histone octamer. Octa means 8, 2 molecules each. 8 molecules of histones, 2 each, H2, H2B, H3, H4, that will carry DNA wrapped around like this, twice, 2 turns, and nearly it's 146 base pair long. And you can see after this, it goes to next histone octamer, then next. So we call this a histone octamer plus 146 base pairs of DNA as single nucleosome. And a nucleosome is considered as fundamental unit of chromatin or chromosome. Okay. And the DNA gets wrapped around these nucleosomes. These nucleosomes, you can see it's like becoming helical. It's becoming more and more supercoiled. Eventually, you see such a structure, which is called chromosome. And this part is the centromere, the central part of chromosome. Then the 2 termini is called telomeres. So that's how DNA is present or packaged within a cell. In case of eukaryotes, you have this nucleosomal packaging of the DNA. This DNA in nucleosomal form, it undergoes, supercoiling. Then there are loops. If you look at the structure of a chromosome or structure of DNA, the way it is packaged within the cell, what we have learned is that, in a human cell, for example, the total length of the DNA. So we have 46 chromosomes, I said. On 46 chromosomes, if you linearize all the DNA of 46 chromosomes, what we will see is that the length of the DNA on 46 chromosomes is 2 meters long. And do you know how big is the size of the nucleus? Is 10 micron. Can you package 2 meter long thread in 10 micron size? It's close to impossible. I think it's just like somebody asked you to fit a grapefruit within a lemon. Physically, it's impossible. So nature has devised a very specialized packaging strategy, and this is how DNA is packaged in a eukaryotic cell. Similarly, the prokaryotic cell also have, supercoiling and, but it's not in the form of nucleosomal packaged material. So if we now go and try to look at how the genome, this is the simplest genome, for example. If we pay attention here, this is E. coli genome. As I said, E. coli genome is a one large circular chromosome. You can see it's nearly 5 million base pairs. And then you have these red and green, what we call the colors. So these colors represent specialized features within the DNA, which are linked to specific genes. So this length of DNA contains nearly 4,200 genes. So bacterial genome is small. Bacteria requires just 4,200 genes to survive and to grow. All the 4,200 genes, they can be transcribed. These are basically coding genes. When I say coding gene means this particular gene is decoded into a messenger RNA and then into a protein. Okay. How DNA molecule looks like, you all must have learned in last week. So DNA is a double-stranded structure. It's a helical molecule. You can see here if you pay attention. So this goes like a helix. Okay, if I draw a central line as a what we call the axis of the molecule, so the strands are going in twists and turns, and both the strands are held together through hydrogen bonds, and these hydrogen bonds are actually between 4 nitrogenous bases. The building blocks, the real building blocks of DNA are actually these 4 nitrogenous bases, which are called guanine, cytosine, thymine, and adenine. So adenine always pairs with thymine, guanine always pairs with cytosine. In simplified form, we have this, ladder-like structure, but DNA never looks like this. DNA is actually a helical structure like this one. Okay. Now, guanine and cytosines, they carry 3 hydrogen bonds between them, and adenine and thymine, they have 2 hydrogen bonds. Why it becomes a helical structure, and why we have these 5 and then 3 and 5 and 3, what actually these 5 to 3 means? What this ribbon, so if these are the nitrogenous bases, what is this red ribbon then? So This red ribbon, we call this sugar phosphate backbone. What is actually happening that you have, we are talking about DNA, and DNA carries, deoxyribose sugar. This sugar molecule has, so here, for example, you will have phosphate. Then on this side, you will have these, nitrogenous bases. So here with this deoxyribose sugar, you have nitrogenous base attached. And on the fifth carbon in this pentose sugar, you have the phosphate group attached. Together, so deoxyribose plus phosphate molecule plus nitrogenous base, together these three are called a nucleotide. Nucleotide. Now, DNA, we said, will be, it's a polymer. DNA is a polymer. It's a molecule which is made up of many nitrogenous bases. How one nitrogenous base is attached to the next, or how one nucleotide is attached to the next. For example, I draw here. You have here, for example, you have phosphate group. You have here, let's say, guanine here. Now, this is one nucleotide. How different nucleotides are attached together? Because we said it's a polymer. They are attached together. Here, this hydroxyl group is bound by the phosphate group of the next nucleotide. Then the nitrogenous base can be adenine. The hydroxyl group here will be bound by phosphate of next nucleotide, and here you may have a thiamine. What is the, so this is the position 3 and this is 5 in 1, 2, 3, 4, 5. Okay, the position of carbons. So what happens, whenever we write DNA, you see here we write it like 5' and 3', 5' and 3', and here also 5. You keep going like this, and its other end will be 3. And on this side, you go from 5' and you end up with 3. Why 3? Because after when chain elongation stops, you have a free 3' hydroxyl group where further elongation of DNA can take place. So DNA synthesis or DNA elongation of a polymer always follows 5 to 3 orientation. Okay, so that's why we write, whenever we write in simplified form, we write like this. Now the question is, why we have 5 to 3, then why the opposite strand? So G, here you will have C, A, here will be T, T, here will be A, so double bonds, here 3 bonds. The DNA is an anti-parallel, it exists as a molecule where 2 strands are in anti-parallel orientation. Anti-parallel means you will never have 5 to 3 and 5 to 3. It's impossible. It will always be, if one strand is this one, the other is opposite. 5 to 3. And elongation is always going to be 5 to 3 orientation. Why it becomes helix? Like you can see here. It becomes a helix because of the angles on which, so the way the next nucleotide is added, and there is a specific angle between this pairing and the next pairing. And that's how naturally there is very strong physical basis. There's physics behind why you see bases are stacked in between. You can see bases are in between, and then you have this sugar phosphate backbone. Now you understand what sugar phosphate backbone is. So these are the bases, nitrogenous bases, and this is that ribbon, sugar phosphate backbone. And we write it 5 to 3. Any question till here? यहाँ तक clear है?
Yes, sir.
Good. So that's how DNA becomes such a double helix. Then I told you DNA is eventually, in case of, for example, in eukaryotes, it's packaged in nucleosomal form. But what is the fundamental unit of DNA is a single nucleotide. And when many nucleotides get connected with each other, that is basically DNA synthesis or elongation of DNA polymer. And when we write DNA, we normally don't write sugar phosphate backbone. We will write like sequence of one of the strand. For example, अगर ये 5 prime थी, ये 3 prime है, we will write, for example, G A T, G A T. It's, and इसकी orientation you 5 to 3 कर देंगे, A C A G A T A, and it's understood. आपको पता होना चाहिए कि ये DNA sequence है, तो you will have a complementary. इसका complementary आप खुद से लिख सकते हो, T A T C T G T A T C 3 prime. जी, there was a question. जी हमजा.
सर मैंने पूछना था कि डीएनए का जो नेगेटिव चार्ज है, लाइक इज इट डिफ्यूज्ड और इज इट जस्ट एट द 5 प्राइम एंड, वो किस तरह लाइक स्प्रेड हुआ होता है उसका?
तो द नेगेटिव चार्ज किस वजह से है डीएनए का?
फॉस्फेट ग्रुप की प्रेजेंस की वजह से.
या सो इज फॉस्फेट in one particular position, yeah, is it spread all over the DNA?
It's spread all over the DNA.
So when DNA will be wrapped around histones, histones are highly basic in nature. तो वो कहते हैं कि negative charge of DNA and histone का जो positive है, because they're excessively basic amino acid, तो वो wrapping में भी मदद देता है. तो डीएनए का जो चार्ज नेगेटिव है वो किसी एक जगह पे कन्फाइंड नहीं है, ये तो थ्रू आउट रैप हुआ इन अ चेन. क्लियर?
ओके, थैंक यू.
गुड. सो नाउ लेट्स लुक एट द डिफरेंट फिनोटाइप्स विद इन बैक्टीरिया. हमने कहा था जीनोटाइप 2 फिनोटाइप. हमने डीएनए को थोड़ा सा देखा है. वी हैव ट्राइड टू अंडरस्टैंड व्हाट डीएनए इज एक्चुअली. ओके. And why it is important, because the information stored in the DNA is decoded and then it results in a living molecule, a living organism. For example, here you have variety of bacteria. You have cocci, you have bacilli, and you have cholera causing bacteria, Vibrio cholera. It's flagellated. Bacilli are rod shaped. You can see जो streptococcus है, you see it's also like a rod but circular है. Cocci are just like single individual cells or maybe in a colony form. How all this is possible? All this is possible because each of this bacteria, it contains very specialized characteristic, its own specific DNA sequence. And that sequence ensures that the information stored in that DNA ensures that cocci remain cocci and it produces cocci. Bacilli, they remain bacilli, they grow and they produce bacilli. Or Vibrio, they produce its own kind of Helicobacter pylori. This is the bacteria जो अल्सर cause करते हैं. आप में से कुछ ने सुना होगा कि डॉक्टर्स रेकमेंड किसी को बहुत ज्यादा एसिडिटी हो या स्टमक में पेन हो तो डॉक्टर से कि अच्छा आप एच पाइलोरी का अपना टेस्ट करवाएं. That is H. pylori is this bacteria which causes ulcer. Now we did talk about genome and then briefly we touched about variation. Early on we said that hereditary is a concept which ensures continuity of the line. Humans always will have babies, cats will have kittens, dogs will have pups, and so on. But we do see variation in the genome, in the phenotypes as well. For example, if you pay attention, even within your family, two siblings, they don't look 100% alike. Am I right? Am I right? Are you there or you are just having your...
Yes, sir, you are right.
So somebody has, flat nose, the other has very sharp nose, eye colors may be different, height may be different. All this is called variation. So in contrast to the hereditary concept, heredity, the other concept is variation. Variation means differences, slight differences in the genome. They are decoded in the form of differences within siblings. Here, for example, you have variation. Here, look at this beautiful variation. This is my favorite one. And this one is even more favorite. You have citrus. And, ये तो different strains हो गई. लेकिन same plant पे आपको different types of fruits नजर, different kinds of fruits, not kinds, different shapes and sizes of fruit. कोई बड़ा हो सकता है, कोई छोटा हो सकता है, ठीक है, कोई ज्यादा sweet होगा, कोई sour होगा. So this is all due to variation in the genotype. There is slight variation in the genome of, let's say, my siblings, which make me different in my phenotype. My appearance become different. And that concept is called variation. Without variation, we cannot do biology. We cannot do molecular biology, DNA science जिसे हम कहते हैं, जो हम लम्स में करते हैं या दुनिया सारी कर रही है. So variation is very important because variation helps us understand different traits, characters, their inheritance. And this is a very common kind of example जो आप सबको पता होगा. Look, blue eyes and then brown or black eyes. All the genes which are responsible for making an eye structure, lens, vision, whatever you name it, they are same in both the individual. Consider this as Pakistani eye, consider this as a European eye. There is variation in the eye color gene. Baki 100% same. Similarly, if you look at here, the skin color of these dogs, they are dogs. 100% सेम, बट उनका जो स्किन कलर है उसके जीन में कोई वेरिएशन होगी. हेयर कलर, look, these are more वो Ethiopia या African hairs are more curled as compared to Asian and European जो more straight या silky होते हैं. And then you can see अगर आप continental spectrum एक बनाने की कोशिश करें from America all the way to Africa, you can see variation in the genome, which is causing difference in appearance in the structure of the hair, kinds of hair, skin color, nose shape. Look, in Africa we have more flat noses as compared to Europe or Asia, where the nose are more sharp. All this is the link between genotype to phenotype. I give you a very interesting example which is coming here. So we all have simple beginning, be it a flowering plant, a mouse, or a desert plant. The sexually reproducing organisms, where male reproductive cell, which is the sperm, and the female reproductive cell, which is the oocyte, they fuse. They all in the early stage look same, very simple beginning, but eventually they become different. This journey is called genotype to phenotype relationship. Genotype is different of this particular species, then this one, then this one. Eventually, that collection of genome or genes is decoded in such a manner that the phenotype becomes very distinct. We no longer remain the same. Although we all at this on day one, we all may look similar kind of cell. Okay. So why we become different? As I told you, we have certain set of genes, collection of genes in our cells, and the information stored within those cells in the form of genes, they become they are decoded into proteins. So which means the functional molecule in a living cell is protein. If I have in a single cell, in a human, if I have 46 chromosomes, there are nearly 20,000 genes on these 46 chromosomes. Each gene, depending upon eye cell, liver, kidney, whatever, they are decoded, and each gene is decoded multiple times. It means protein is being made eventually. The collection of all the proteins, which I refer to as proteome, in a living cell or in an organism, they are the ones which differentiate us from one another. DNA and proteins, they make the hardware. DNA is the one which has stored the information, but the real functional molecule in a living cell is the protein. And based on our differences in our genomes, we have eukaryotes, we have these prokaryotes, then plants, and then bacteria, photosynthetic bacteria as well. And then there is a third tree between prokaryotes, eukaryotes, and then there's a third branch of tree which is called Archaea. They are similar to bacteria, but their machinery is more like eukaryotes. How we draw these, this tree is basically, this tree is called phylogenetic tree, where different species are compared with each other using their DNA, and then they're based on the similarity in different genes and proteins and their collection of genes. This tree is drawn, which is called, basically it tells how far evolutionary, how distant one species from other. But these differences in the form of phenotype, it's very clear. It's due to the proteins and the underlying genetic information which is stored in the DNA. Now, in last century, when it was not clear that DNA and protein are actually linked to one another, DNA is the molecule which stores information and protein is the decoded form of that information. There were people working with, scientific community was working with proteins and DNA. There were scientists who were working with nucleic acids, which was DNA, RNA, etc. And there were scientists who were working with proteins. Now, there was a big question. The ones who were working with nucleic acid or DNA, they were saying the molecule of heredity is DNA. And the ones which were working with protein, they were saying it's the protein. Now, how do we prove, is it the DNA or the protein which determines, or which is the molecule of heredity, which is responsible for inheritance of traits and characters from parents to offsprings? And it's a beautiful electron micrograph if you pay attention here. This is a virus called bacteriophage. Bacteriophage. The one which kills bacteria. This is a bacterium. Look, this is a bacterium. And this is a phage captured infecting the bacterium. Virus by itself is a dead entity. It's very interesting thing. We don't categorize it as a living molecule or living cell because it cannot reproduce, it cannot replicate itself at its own. If I leave virus on my table, purified virus, it will remain there forever unless it gets access to a living cell, which it can infect. And then once it goes within the cell, then you know what it does. It does what COVID is doing with us. Okay, so bacteriophage is a virus which infects bacteria. Why we are suddenly talking about viruses and bacteria? Because we want to answer the question, which molecule, DNA or protein, is hereditary molecule, which is responsible for passing on traits from parents to offspring, from mother to daughter cells. Okay, so two very intelligent scientists, I think they are Harsha and Chase. Yeah, they came up with a very interesting experiment. They knew about bacteriophages. They were working with bacteriophages and bacteria. They knew that the bacteriophage has, a head, which is made up of proteins. Within this head, there is the genome of virus, a DNA, a certain length of DNA is packaged. Then you have the neck, this collar, and then you have these tails made up of these proteins. All these structures of virus, they are encoded by the DNA within the bacteriophage head. Now, they also knew that when virus infects bacterium, within the bacterium, now it can replicate itself and eventually it bursts open, it kills the bacteria. So virus infected bacterium. This bacterium, so now all these are viruses being replicating, okay? One virus infected, and then they knew that eventually this cell dies, it bursts open, and then all the viruses are released. And they move on to infect the next bacterium. Kill this one, move on to next bacterium, and keep growing once they have found the host. Now the question they said, this is a very interesting model. We have head, collar, sheath, etc., these tail fibers, and we have the DNA molecule. Why cannot we use this to answer the question if it is the DNA or the proteins which is hereditary molecule, which parents pass on to their offsprings or next progeny, or one cell to next? Now, they came up with this idea because they had very good fundamentals and they knew that the DNA molecule, it contains sugar, phosphate, and what else? Nitrogenous bases. And protein, it contains, प्रोटीन किसकी बनी होती है?
Sir, amino acids.
Amino acids. Very good. So proteins are made up of amino acids. And they knew that the first amino acid, I'm saying first amino acid in a majority of the proteins is methionine. And this methionine contains sulfur. There is another amino acid called cysteine. Cysteine also contains sulfur. Okay. Now they came up with a very interesting idea. They said, we are going to grow viruses in a medium where we will have, let's say, bacteriophage. We will grow this in the presence of medium and bacterium indeed, where we will add medium with radioactive phosphate. Is it clear? And then we will also have phages, bacteriophages, in which medium will contain sulfur, radioactive sulfur, S35 radioisotope, and P32 we call this. Okay. Now, what will happen that the bacterium which will grow in the presence of radioactive phosphate, it's all the DNA will become what? DNA में क्या होगा? P32 radioactive isotope incorporate हो जाएगा. And the ones which were grown in the absence of radioactive phosphate but in the presence of sulfur isotope, उसकी जो proteins हैं उसमें, and what are the proteins? ये head वाली protein हो गई, sheath, collar, tail fiber, ठीक. इनमें S35 incorporate हो जाएगा. And now what we are going to do, we are going to use the phage which was grown in the which was grown in the presence of P32. We will add all this in a bacterium culture, and separately all the virus which was grown in the presence of S35 in a separate culture in bacterium. You let them infect for some time, 10 minutes, 15 minutes, whatever. And then what you do, you blend this virus plus bacterium. So इसमें क्या होगा? इसमें virus must be attached to bacterium. Here also virus must be attached to bacterium. You put everything in a blender and full speed पे उसे blend कर देंगे ताकि ये connection breakdown हो. Virus and bacterium are separated. And then you spin all this mix in a centrifuge. Centrifugal force will apply, and the heavier particle, which is bacterium will settle down. And viruses are क्योंकि बहुत ही microscopic है, much lighter, they will keep floating in the, इसे हम ऐसे draw करते हैं. All the bacterium will settle down due to high speed centrifugation. उसे यूं आप rotate करते हो due to application of high centrifugal force. So you rotate like 14,000 RPM पे से rotate करते हो 5 मिनट या 2 मिनट. सारा बैक्टीरिया सेटल कर जाएगा और ये जो लिक्विड रह जाएगा we call this supernatant. इसमें सारा वायरस होगा. Same will happen here. You will have बैक्टीरिया सारा सेटल कर जाएगा और वायरस इस पे हो. What they wanted to see bacterium जो मैंने grow किया है, is it P32 or is it S35? It contains P32 or S35. Why? Because if the molecule of heredity, which is, parents pass on to offsprings, if that is a DNA, then virus must inject only the DNA. And inside the bacterium, when I settle down through centrifugation this bacterium, I should find P32, which is basically the labeled DNA of virus. In case it's not the DNA, protein is the hereditary molecule, then when I spin it down, I should see all the S35, क्योंकि amino acid ने proteins को किया होगा. अगर protein inject किए हैं बैक्टीरिया के अंदर वायरस ने, तो फिर मुझे S35 अंदर नजर आनी चाहिए बैक्टीरिया. When they observed this, they discovered that it's the DNA, क्योंकि जो S35 है, it all remained in the supernatant, but all the P32, it went into the pellet inside the bacteria. So that was a proof that the molecule of hereditary is basically a DNA molecule. Any question till here?
No, sir.
So ये वही हर्शे और चेस का जो experiment है, that concluded that the information which is required to make more copies of our virus is stored and hidden inside the DNA. So they not only prove that DNA is the hereditary molecule of inheritance, but it contains all the information as well, which is required to produce more of its own kind. Because after infecting within the bacteria, we could see when it killed the bacteria, many more viruses are released. It means the DNA which was injected, it contains all the information for life of virus or any other organism. So how this information is stored, I already told you. Information is stored in the form of nitrogenous bases. We have A, T, C, G, all living organisms from bacteria to humans. This is the way how information is stored within DNA. Now, in human, but from species to species, this the amount of information stored may vary. For example, and the number of these bases definitely vary. For example, in humans, we have the 6.4 billion base pairs in a diploid genome. Diploid genome, 2N. Diploid means where each chromosome has its homologue or copy. This is human genome. 46 chromosomes and 22 pairs, 44 okay, 45, 46. We will talk about what X and Y are in our later lectures. Now, there are 6.4 billion AT, CG, CAT, TAT. These are just 4 bases repeated again and again and again. It looks very useless, you know, that 4 alphabets are repeated, but actually they are repeated in a very, very systematic and a specific manner. It's not just random. If I use word random, it's wrong. It's a very specific order, which, so let's say ATG, this one encodes an amino acid which is called methionine. I have CAG, this one encodes a amino acid which is called glutamine. So there is a very specialized order of the arrangement of these nitrogenous bases on the chromosomes. And all the information is actually stored in the DNA in the form of nitrogenous bases. I told you, I explained you already the sugar phosphate backbone. And I also told you that this ATCG, CAG, whatever, these are not just random nucleotides. There is a specialized order in the sequence. As I said on this slide, that, you know, most of the time, the first amino acid is methionine, which means you have to have a codon. So इसे एक को triplet sequence को ATG को हम codon कहते हैं. We will talk about codons in the genetic code in more detail in next lecture. But what you should know is that all the information which is responsible for reproduction of an organism and making its own kind is stored in the DNA. Even not only at the organismal level, at cellular level. Let's say we were babies in our mother's womb on day 1. We were single cell after fusion of sperm and the egg. This went to 2 after cell division. They divided and they became 4. They divided and now we have how many cells? 8 cells. This cell division and this growth from mother cell to, you know, all this progeny of daughter cells, this is not possible without DNA molecule. All the information is stored within the DNA molecule. Any question? So now we are going to talk about how DNA molecule is copied. And this DNA molecule copying copying DNA molecule means basically, you know, you have a double-stranded DNA molecule and from one molecule we have to have 2 molecules, because without this process, 2 identical molecules, because without this process, from single cell, you cannot become 2 cells. life cannot continue. You cannot grow, whether it's a development of tissue or organism or whatever. So you have to replicate. This process is called replication. Replication. In case of DNA, we call this DNA replication. So how this copying mechanism takes place, we are soon going to see, because without this, neither you can grow, you cannot pass on information to the progeny. But this understanding of this process also helped us understanding another big question, which was how the information is decoded. These are two different questions: how DNA is copied and how information is decoded. First, we are going to see this cartoon, which shows you the DNA replication. You can see this circle with an arrowhead. It shows you DNA molecule copying itself. Okay, and then the flow of information is DNA, information stored in the DNA is decoded into an RNA molecule, ribonucleic acid, and this RNA molecule is further decoded and a functional molecule, which is protein, is made. So this is what I refer to as earlier central dogma in molecular biology. Okay, this is taking place in all living organisms, bacteria to fruit flies to fish to humans to plants, mouse, animals, etc. In bacteria. So that's what the conservation in evolution, from simple most organism to most complex organism. And we also, in the same way, we also talked about some variation. Even that is happening because flow of information, same gene which is responsible for eye color. Here, one individual will have this where there is slight variation. So end protein will result in a blue color eye or black colored eyes. Now, how this process takes place, how DNA is able to replicate itself, how it can go from here, single molecule to two molecules. There's a dedicated machinery. So the enzymes which copy DNA into DNA are called DNA polymerase, and it's a dedicated machinery. We call this DNA replication machinery. We are going to look at it in detail. Then the enzymes which decode information stored in DNA into RNA, they are called, so enzymes which make an RNA by copying DNA, they are called RNA polymerases. And the proteins which make a protein by decoding RNA, they are called ribosomes. So the process, this process, decoding DNA into RNA, is called transcription, and this is led by proteins called RNA polymerases. The process decoding RNA into protein is called translation, and it's led by ribosomes. It's a huge protein complex. And the process of DNA replication is led by enzymes called DNA polymerases. Okay. Now, this one is the central dogma. Okay, but there is also a exception in this flow of information. There are organisms, viruses, which do not have DNA at all. They are just RNA molecules. And I don't need to tell you which virus has RNA, which is hovering around all of us, and that's the COVID. It does not have DNA. It has just RNA. When it gets into body in its host cells, it makes its proteins and it replicates itself, makes more viruses. There are viruses which are RNA, but they convert RNA into DNA, DNA back into RNA, into protein, and then viruses. Such viruses, an example is, for example, HIV/AIDS virus. It's called retroviruses. Retro because they're going in reverse transcribing their RNA. Their natural genome is RNA. Okay, so from RNA they make DNA, and then they can replicate. That was all about different processes. Now let's look at the DNA replication. This is why DNA replication is important. This is a single cell. It's undergoing DNA replication. Look, it was single cell. It's copying all its genome. Every nucleotide, every base pair in the genome is duplicated. It's being copied. And look, now let's say this was a one big circular molecule. Now you have two circular molecules. Cell divides into two, and you have two identical cells. You had, let's say, one circular molecule in this bacteria. DNA, due to DNA replication, now you have the two, and they are passed on to two daughter cells. If you cannot properly execute DNA replication, you cannot grow as an organism. You develop defects, you may die. Remember, I told you DNA synthesis always occurs 5:3. I told you why. And protein synthesis always occurs amino to carboxyl terminal. Okay, you have this is like, let's say, a simple amino acid. You will always have amino acid with, so DNA synthesis 5:3. I told you, you have a free hydroxyl group at third carbon, where phosphate of the next nucleotide will come, bind, and polymer will be synthesized. Proteins are amino to carboxyl terminal. So you have, for example, this is the amino group. This is one amino acid structure. Okay. At the carboxyl group, you will have new peptide bond will be made. So we will lose the hydroxyl group, and we will have the amino group of the next amino acid will come, bind here. You will have next amino acid attached here. At the carboxyl group of this amino acid, the next amino group of the next one. So we call this peptide bond. Peptide. So this peptide bond is between carboxyl group of one amino acid and amino group of the next amino acid. That's why in case of DNA, we have 5' to 3' end. In case of protein, so this is DNA. In case of protein, we say N terminus to C terminus, or amino to carboxyl terminus. G Mason?
Sir, DNA synthesis और replication में क्या difference है?
DNA synthesis or replication में क्या difference है? Just wait, it's coming on next slide. Okay. So this is process of DNA replication. So DNA replication will involve DNA synthesis. Okay. But every DNA synthesis may not be DNA replication. We may be synthesizing DNA in a machine. That may not be DNA replication. But DNA replication definitely needs DNA synthesis, which will be present on this slide. So based on experiments of Michelson and Stroll, I will highly recommend you to go and read this experiment at your own in detail. It's a simple experiment. They proved that DNA replication, because before their conclusion, before their groundbreaking work, there were 3 models. One model used to say that DNA replication is conservative. The other one used to say it's semi-conservative. And yet another one used to say it's dispersive. Okay, dispersive, conservative, and semi-conservative. Now, if you pay attention to color code, this is the parent strand, parent molecule, and this is after DNA replication. Conclusion of Michelson and Stahl was that DNA replicates following a semi-conservative mode of replication. What happens that both strands will be copied. One complete new strand will be synthesized copying one of the mother strand. Like here, blue one is the mother strand and new strand is being synthesized. In case of conservative, they were saying, no, both the strands remain intact, but they are copied and daughter strands are 2 independent daughter strands are making 1 molecule of DNA. And dispersive was that, you know, there are some, so the old strands, both the old strands, they will be dispersed by newly synthesized pieces of DNA. This was quite interesting, but I fail to understand how they could, you know, think of this. But Michelson and Stahl proved, and this is what we all believe till today, that DNA replication follows semi-conservative mode of DNA replication. What it means, actually. We learned in the earlier part of the lecture that DNA synthesis takes place from 5' to 3' end, and we learned that there is this sugar phosphate backbone. This hydroxyl group of 1 nucleotide and the phosphate group of the next, they are linked together through what we call phosphodiester bond. Okay, it's a chemical bond, and that's how sugar phosphate backbone is established. So if you have this 5' to 3' strand and you have to copy this, Michelson and Stahl said that you have to have a free hydroxyl group, and then there should be an enzyme which brings in a new nucleotide which is complementary to this one, which must be cytosine here, together with its sugar phosphate, and make a phosphodiester bond. After this nucleotide is attached, like here, it's attached now, you have the free hydroxyl group, new nucleotide will come, which will be a guanine with its sugar phosphate backbone, with its sugar and phosphate components, and then you will have again here phosphodiester bond, and then you will have chain elongation or mesom, what you were asking. This is actually DNA synthesis is taking place, but since we are copying a pre-existing strand, we call this DNA replication. The end result is you have, let me draw here, you have five to three. This means the one mother strand, which was this one, top one, and the bottom one, they got 5 to 3, and you have to synthesize their copy. Okay, and here 5 opposite is always anti-pair. 5 go 5 to 3. Only then we will have the daughter strand synthesized. मैंने rectangle इसलिए बनाया कि ये daughter strands है, ठीक है, just to distinguish them Now the question was how the synthesis starts. It was very simple what I said. I said you have 3 to 5 strand and then जो है ना daughter strand start हो जाएगी. It's not that simple. Nature has engineered very specialized manner. DNA polymerase, the enzyme which synthesizes DNA, it cannot start DNA synthesis de novo. De novo means from scratch. It requires a free hydroxyl group. So we don't have here a free hydroxyl group. Are you able to understand what I'm trying to explain you? Let's say we have G here, we have to add cytosine here, but where to add cytosine? We don't have a free hydroxyl group. What we discovered in last 50 years, people learned that when DNA replication starts in a living cell, you know, it requires a primer. And there is a very, very specialized protein machinery, which is called primase. It's an enzyme. What it does, whenever DNA replication starts, because the point at which DNA replication has to start, it's encoded by DNA. This information is present in our DNA, in bacteria, in humans, in all living organisms. At that particular point, this primase comes in and it adds a small RNA molecule. We call this RNA primer. And look what it has done. De novo, it has made an RNA molecule. It has 5' and 3' free primates. Now will come the DNA polymerase 3. It has acquired the hydroxyl group. यहाँ पे OH होगा ना? So DNA polymerase is going to add now nucleotides. And it will keep going and it will make a complete DNA strand. Sorry. So here, sorry, you have ये hide हो. आपको नजर आ रहा है ये red वाली जो है या ये hide हुआ है मेरी. Are you able to see this?
Yes, sir.
So you can see now a complete, new strand is synthesized, and the process is DNA replication. From one strand of DNA, you have copied another one, but you have the RNA primer is still there. So in case of DNA replication, what happens? Now we have to imagine two strands. In the previous slide, we just simplified कि जी अच्छा single strand यूं होगा. If we had two double-stranded, we have two strands. DNA is a double-stranded molecule. Every living cell, when it starts DNA replication, within the DNA, there are very, very specialized sequences, which is called origin of replication. Just a DNA sequence. वहां पे certain proteins आके bind कर जाती हैं जो ही DNA replication होनी है, and they tell the cell start DNA replication from here. And that in bacteria, in humans, we call this origin of replication. Now, why it is important? Because at that particular point, an enzyme comes. We call this enzyme helicase. DNA helicase comes in and it unwinds. It breaks down. ये जो हाइड्रोजन बॉन्ड्स है ना, ये ब्लू वाले, ये हाइड्रोजन बॉन्ड्स डिस्टरप्ट हो जाते हैं, डीएनए becomes single stranded at that particular place. We call this region is called replication bubble, क्योंकि यूं वो जरा single stranded हो गया. RNA primase is going to come and primase is going to add RNA molecule, and then free hydroxyl group होगा और वहां पे DNA polymerase 3 आके तो strand बनाना शुरू कर देंगे. Now this is very interesting because 5 to 3 and then opposite strand is being made 5 to 3. What we will do with this one? This is 5 to 3. Replication is going in this direction. you cannot synthesize 5 to 3 here. So this strand, which is being synthesized continuously, we call this leading strand. But the opposite strand, which is 5 to anti-parallel strand, we cannot synthesize this. So we replicate this in bits and pieces. RNA primase adds a tiny primer here. It is 5 to 3, और वो DNA polymerase आके इसे करता है. मजीद DNA अनवाइंड होता है, RNA primase यहाँ पे आता है, RNA primer ऐड करता है, and then extension होती है, DNA synthesis होती है. So this DNA is called discontinuous or lagging strand. Okay, because this is in bits and pieces, and these short fragments, ये जो छोटे-छोटे fragments बन रहे हैं, we call these fragments Okazaki fragments. Okay, here, look. Oh, why? Here you have this Okazaki fragment. This is continuously being replicated. This one is in bits and pieces. One Okazaki fragment, then another one. As it will keep opening, another RNA primer will come, and it will keep growing like this. Is it clear so far? Yes or no?
सर जो लैगिंग स्ट्रैंड है वो एक दफा थोड़ा सा एक्सप्लेन करते हैं.
Leading or lagging?
लैगिंग स्ट्रैंड लाइक
Okay, lagging
आरएनए
strand.
प्राइमर जो
Yeah.
RS पे.
So lagging strand is, why it is lagging? Because it's not being synthesized in a continuous manner. So this is continuous because we learned DNA synthesis takes place in 5 to 3 orientation because of availability of free hydroxyl group. In this case, orientation of the parent strand is 5 and 3. DNA replication तो इस डायरेक्शन में हो रही है, in this direction. तो आप यहाँ पे 5 to 3 तो नहीं ना कर सकते, इसका opposite तो 5 to 3 anti-parallel तो यूं होगा. Clear? So what happens that RNA primase adds a short RNA primer. It synthesizes what you call the DNA strand, which gets connected with the next Okazaki fragment. जब यहाँ पे मजीद इस डायरेक्शन में replication bubble expand होता जाएगा, क्योंकि ये DNA इस डायरेक्शन में, you can see this arrow, it's unwinding in this direction. As soon as these, so here you can see this RNA primase is already sitting here to add now next primer, and then DNA polymerase 3 will extend. तो ये इस तरह bits and pieces में, fragments में extension होगी या DNA synthesis होगी, इसलिए इसे हम lagging strand कहते हैं. Is it clear?
Yes sir, thank you.
Okay. So this is just a quick summary of DNA replication. What has happened? You have DNA unwinding के बाद just एक strand आपको दिखाई जा रही है, which is basically in case of discontinuous or the lagging strand. Here you can already see a Okazaki fragment. ठीक है, ये था DNA polymerase ने कर दिया. Primase again added another RNA primer. DNA polymerase 3 is going to now synthesize, which it has done here. And then what eventually you have RNA primer, DNA sequence, RNA primer, DNA sequence, RNA primer, DNA sequence, and then the mother strand. Now the puzzle is, you cannot keep RNA sequences within DNA. You have to get rid of them. So there's another DNA polymerase, which is called DNA polymerase 1. It comes in, it hydrolyzes, it destroys the RNA molecule, okay? And it synthesizes, in place of RNA, it synthesizes then the DNA sequence. And finally, an enzyme called ligase, it comes in and it joins these यहाँ पे शुगर फॉस्फेट बैकबोन बना देते हैं। तो इस तरह ये भी पूरी एक स्ट्रैंड बन जाती है। None of you asked me a question कि is there possibility of an error, and it's just an enzyme is reading, for example, here, for example, an enzyme is reading, you know, in front of A should be T, G, in front of G should be C, in front of T should be A. But the enzyme is just adding. So is there a possibility of an error? Yes, there is possibility of an error. These enzymes, DNA polymerases, they can add mismatch or an error. So instead of T in front of an A, it can add G or C. So we call this mismatch or an error. And they say the possibility or probability is 1 in a million, I believe. Okay, 1 in a million, 10 to power minus 6. No, I may be 1 in a billion. Abdullah, is Abdullah there? So I will confirm this. Is it 1 in a million or 1 in a billion? Whatever. These enzymes can add error, but these enzymes also have an activity which is called proofreading activity. As they move on, if by mistake they added a mismatch, they will stop, read, and then they have the ability to remove it and bring in the correct complementary nucleotide and then keep synthesizing. So there are that was proofreading enzyme, proofreading activity. But in case of DNA, there are other possibilities as well that, you know, we may get DNA damage. We may get or jab DNA damage, for example, if somebody is smoking too much, if somebody is alcoholic, or, you know, you're under tremendous stress, so your body, your cells start producing reactive oxygen species, which are very dangerous for DNA. They can damage DNA. When DNA gets damaged, DNA tries to repair itself, because nicks or large breaks or small breaks or single nucleotide mismatch, they are very dangerous, and cell immediately tries to repair it. उसे repair तो कर लेता है, but repair machinery then tries to see if it is a mismatch, it is a single nucleotide, large nucleotide. तो उसकी एक पूरी repair machinery है. That's a different subject. But in case of DNA replication, if enzyme finds there is a mismatch, like here I showed you, it proofreads, and after proofreading, there is a specific activity in that enzyme. It can remove just this one single nucleotide, or it can remove a chunk of DNA neighboring nucleotides together with mismatch, and we call this one excision. Excision means it excise and remove the whole small piece together with a mismatch, and then DNA polymerase repairs or corrects the this mismatch. In case of mismatch repair, what it does normally is that it does not normally remove everything. If it has the mismatch repair protein excise the a chunk of DNA together with the error, which was a nucleotide which was wrongly placed, it will fix this nucleotide here and bring it back into the position from where it had removed. So DNA polymerase adds then the correct bases. So in case of mismatch repair, idea is to just fix mismatch of a single nucleotide. In case of excision repair, it excises and then DNA polymerase adds the correct bases. Just like in case of DNA replication, it synthesizes by going from 5' to 3' orientation. Okay. How much time I have?
So 10 minutes.
10 minutes. Yeah, we are just at the end of our lecture. Yeah. There is one more thing which should come to your mind, that when you are unwinding DNA, you know, oh yeah, this is good. Can you see this cable? Telephone की cable देख रहे हो?
Yes, sir.
Yes, sir.
So I just looked around to explain you something, and this is what I found, and I thought this is the right helical structure. Imagine this as a double-stranded DNA. And what you are doing, you are actually replicating this. This double-stranded DNA is being unwound, and unwinding is going in, let's say, in this direction. So by unwinding, what you are going to do, you are going to zip, and what will happen, if you have ever dealt with the telephone cable, you may have seen it gets, you know, super coiled and like इस तरह हो जाती है ना? सर हमारे पास तो मोबाइल फोन है, हमने कभी इस तरह का फोन यूज ही नहीं किया. Is this the answer? Have you ever seen phone cable? So अगर ये यूं है straight, तो ये ऐसे सुपर कॉइल हो जाती है, ट्विस्ट हो जाती है. होती है कि नहीं होती? This can happen when you are unwinding DNA, because you are pushing. आप एक जगह से उसे पुल करते जा रहे हो, तो ये सुपर कॉइल्स आगे जनरेट होते जा रहे हैं. And this can be a stumbling block. This can simply block the DNA replication bubble if we don't solve this problem. And when you undergo DNA replication, there are specialized proteins or specialized enzymes which are called Topo Isomerases. These Topo Isomerases, here it is, okay, Topo Isomerases. They are the ones which actually move ahead of here, and they are unwinding not the two strands. They are trying to keep this entanglement or entangled supercoil to the minimum so that DNA replication goes smoothly. And these are very fascinating proteins. They can create just a nick, just एक single strand को सिंगल स्ट्रैंड का शुगर फॉस्फेट बैकबोन जो है ना, वो डिटैच करके उसे अपने पास ही रखेंगे. ठीक है, दूसरी को उसमें से निकाल लेंगे जैसे यहां पे हो रहा है, यू कैन सी, एंड देन उसे रीलाइगेट कर देंगे. एंड दैट्स हाउ दीज 6 बिलियन बेस पेयर्स इन डिप्लॉयड सेल्स, जो कि यहाँ पे इन क्रोमोसोम पे हैं, ये सारे आपने रेप्लिकेट करने हैं. सेल साइकिल डॉक्टर फैसल ने पढ़ाया है.
Yes, sir.
Yes, sir. Yes, sir. The M phase.
Yes. So whatever I told you, that was happening in the S phase. DNA replication यहाँ पे हो रही है. In a human cell, you have 18 hours or so to replicate all 46 chromosomes. 6 billion base pairs in a flawless manner without an error. So that is done through this DNA replication machinery, and Topo Isomerases are there. And after DNA replication, let's say you had here 46 chromosomes. ये जब डिवाइड कर देंगे एक सिंगल क्रोमोसोम, ये वाला फॉर एग्जांपल, ये जो नजर आ रहा है, ये आपको यहाँ पे नजर तो नहीं आएगा, लेकिन इवेंचुअली ऐसा हो जाएगा. Why? A single chromosome, I told you, it contains one double-stranded DNA molecule, double-stranded. तो ये सारा DNA replicate होगा तो इसकी ये sister chromatids बन जाएगी. 46 chromosomes इस तरह बन जाएंगे. After DNA replication, then in this phase, cell ensures that every chromosome is duplicated Now I can enter into mitosis, and I can, from single cell, I can produce two cells, two identical cells, if it is mitosis. So this is what you have already covered in PCR. You have covered in your labs or in your 101 in your oh such ये तो इसमें नॉन एसएससी बच्चे भी हैं. I'm sorry, I forgot. So PCR polymerase chain reaction is basically machine. It's आप सारे कोविड का आजकल सुनते हो कि उसका PCR पॉजिटिव आ गया, उसका PCR नेगेटिव आ गया. PCR is a machine which follows all the principles of DNA replication to amplify DNA or RNA molecule. Okay, and what we do, it follows all the principles of DNA replication, but we eliminate many things. We don't need helicase. For example, this is the piece of DNA you want to replicate. Now, what you will do, you will unwind this. How? You will simply heat. 95 degree Celsius पे heat करोगे, ये दोनों strands separate हो जाएंगे. Now you have the primer. ये आप खुद design करते हो, add करते हो, ठीक है? And these primers will come and bind to their complementary sequence. क्योंकि you get rid of the RNA primase, blah blah blah. This primer provides the free hydroxyl group, and after heating it, यहाँ पे आप 95 डिग्री सेल्सियस पे हीट किया है, यहाँ पे आप इसे 60 डिग्री सेल्सियस पे ले आते हो. 60 डिग्री सेल्सियस पे क्या होता है, ये अनीलिंग हो जाती है. अनीलिंग, बाइंडिंग ऑफ प्राइमर टू टेम्पलेट. यहाँ पे 95 पे क्या होता है, डीनेचरेशन होती है. डीनेचरेशन, अनवाइंड हो जाते हैं टू स्ट्रैंड्स. After annealing, you again temperature 72 degree Celsius पे ले जाते हो, and this temperature is called extension. DNA synthesis की temperature. In PCR, normally हमारी body में 72 नहीं होता, हमारी body में 37 ही होता है. So यहाँ पे आप एक खास enzyme करते हो use, which revolutionized the DNA technology or recombinant DNA technology. That enzyme is called Taq polymerase. It was discovered in Thermus aquatus. It's an QUAI aquatus. This is a bacteria which lives above 75 degrees Celsius. It's an extremophile. So its DNA replication uses this enzyme which is stable. So at 72, what happens? ये free hydroxyl group पे न्यू न्यूक्लियर, क्योंकि इसी एपेंड ऑफ ट्यूब में या इसी मशीन में जो ट्यूब आप यूज करते हो उसे हम पीसीआर मिक्स कहते हैं. एक ट्यूब होती है जिसे हम एपेंड ऑफ ट्यूब कहते हैं, छोटी सी 200 माइक्रोलीटर वॉल्यूम की होती है. तो उसमें हम लोग ये डीएनए रेप्लिकेट हो जाता है. अच्छा, उसके बाद क्या हुआ, लेट्स से दिस इज 1000 बेस पेयर जो आपने रेप्लिकेट करना है. इतने से एक जीन एम्पलीफाई आप कर रहे हैं, ये 30 सेकंड्स में कर देगा. 30 सेकंड्स में ये सिंथेसाइज कर देगा. व्हाट यू विल डू, यू विल टेल दिस, ओके, हीट अगेन नाउ 95 डिग्री सेल्सियस. ये हीट हो जाएगा, ये सेपरेट हो जाएगा. अब ये टेम्पलेट बन गया इसका. ये पहले क्या था, ये दो मदर स्ट्रैंड्स ये थे, ये और ये. जब आपने इसे हीट किया, अब जब इसे हीट किया तो अब कितने हो गए आपके पास, 4. ये भी टेम्पलेट है, ये जो नए बने थे ये भी टेम्पलेट है. प्राइमर आके बाइंड करेगा, टेम्परेचर 60 पे ले जाएंगे, 72 पे लेके जाएंगे, एक्सटेंशन हो जाएगी. तो ये आप क्या करते हैं, यू टेल द मशीन, यू प्रोग्राम इट 95 डिग्री सेल्सियस 30 सेकेंड्स, 60 डिग्री सेल्सियस 30 seconds, 72 डिग्री सेल्सियस 30 सेकंड. इट डिपेंड्स अपॉन 72 पे कितना टाइम देना है. अगर आपने 2 के भी लेंथ एम्पलीफाई करनी है तो 2 मिनट्स देने पड़ेंगे. तो आप इसको आप मशीन को कह देते हो, रिपीट दिस 35 टाइम्स, 35 PCR cycles, 30 seconds 30 seconds 30 seconds on these temperatures, 35 times वो रिपीट करता है. Imagine you start from one molecule. After one cycle, you will have two molecules. After second, 4, 8, 16, 32, 64, 128, 256. You exponentially can amplify the DNA, amount of DNA. तो जो COVID वो करते हैं कि COVID जो है वो positive आया है, negative आया है, they are actually making cDNA of COVID, क्योंकि जो COVID है is a RNA वायरस, उसका cDNA बना लेते हैं रिवर्स कॉम्प्लीमेंट्री, फिर उसके स्पेसिफिक जीन्स को वो इसमें एम्पलीफाई करते हैं फॉलोइंग द प्रिंसिपल्स ऑफ डीएनए रेप्लिकेशन, एंड इवेंचुअली दे टेल यू कि यू हैव पीसीआर पॉजिटिव और पीसीआर नेगेटिव. So with that, we finish today's lecture. If there are any questions, please feel free to ask. You can contact me anytime. I'm always there. This is about DNA, is the genetic material, blah, blah, blah. This is about the Dolly, the sheep which was cloned, and even the Jurassic Park. You have seen the movie. This is just to create some fun, but let's leave it. I would like to take some questions. Ji Hamza.
सर मेरा क्वेश्चन ये था कि जो हमारे पास मल्टीपल एंजाइम्स हैं, डीएनए पॉलीमरेज 1, हेलिकेज, पॉलीमरेज 3 एटसेट्रा एटसेट्रा, तो ये लाइक एक्चुअली हमने अभी सेल सारा स्टडी किया था, तो लाइक आई डोंट हैव अ बैकग्राउंड बायो का, आई लाइक टू नो कि लाइक स्टोर कहाँ होते हैं, ट्रांसपोर्ट किस तरह होते हैं, सेल तक किस तरह आते हैं.
They are all within our cell. So DNA polymerase 1, DNA polymerase 3, primase, helicase, Topo Isomerase. Our single cell contains 46 chromosomes, and these 46 chromosomes, each chromosome has genes on it. ठीक है, एक specific length of DNA is called gene. Next जब lecture होगा उसमें explain करूंगा आपको what is gene. तो वो जब डीकोड होता है, उसकी ट्रांसक्रिप्शन होती है, तो हमारा सेल ही टोपो आइसोमरेज भी प्रोड्यूस कर रहा होता है, प्राइमेज भी कर रहा होता है, आरएनए पॉलीमरेज, डीएनए पॉलीमरेज, लाइगेज एटसेट्रा. नाउ सेलुलर मशीनरी इज वेरी इंटेलिजेंट. डीएनए रेप्लिकेशन में जो चीजें चाहिए वो सिर्फ उसी टाइम पे प्रोड्यूस होती हैं, वो उसी टाइम पे यूज होती हैं, एंड एवरी प्रोटीन हैज अ हाफ लाइफ कि भाई अच्छा अब इसकी जरूरत नहीं, इसे गेट रिड ऑफ दिस. नो प्रोटीन इज स्टोर्ड फॉरएवर इन द सेल. when we go through DNA replication, mitogenic signal आता है, DNA replication का signal आता है कि जी अच्छा cell is going to enter into S phase. सारी वो उससे पहले S phase start होने से पहले वो सारी transcription और translation उन proteins की हो जाती है, ready हो जाती है, and then we have process of DNA replication handled very smoothly. अगर उनमें से किसी एक में लिमिटिंग फैक्टर आता है तो वो तो बड़ा डिजास्टर हो जाएगा. तो कहीं किसी डेडिकेटेड प्लेस पे स्टोर नहीं होती. प्रोटीन्स कहाँ पे बनती हैं? फॉर एग्जांपल प्रोटीन्स आर सिंथेसाइज्ड ऑन एंडोप्लाज्मिक रेटिकुलम और इन द साइटोप्लाज्म. प्रोटीन एक खास, हर प्रोटीन की एक खास डेस्टिनेशन होती है. किसी प्रोटीन ने डीएनए उस पे न्यूक्लियस में वापस आना है, ट्रांसलेट होके उसने वहां डीएनए पे बाइंड करना है, या न्यूक्लियर मेम्ब्रेन में रहना है, उसने प्रोटीन शटल करनी है, या सेल मेम्ब्रेन पे चला जाना है, किसी सेल सिग्नलिंग रिसेप्टर से इंटरेक्ट करना है. तो वो सेल्स के अंदर ही ये एक पूरी व्हाट आई कॉल दिस, इट सेल इज अ फैक्ट्री. Cell is a factory which contains enormous number of machines working in real time. As I'm talking to you, I'm utilizing I don't know how many ATP molecules. So and all my body is supporting me. So ये सारी proteins वहाँ पे बन रही हैं, energy synthesize हो रही है, मैं आप लोगों को देख रहा हूँ, vision बन रहा है. So it's a very fascinating thing. अगर आप इसे एक cellular level पे देखो और इसे आप पढ़ो तो आपको पता चलेगा कि अच्छा ये प्रोटीन्स डीएनए रेप्लिकेशन के बाद अच्छा ये डिसअपियर हो गई हैं कि भैया अब तो इनकी जरूरत नहीं है ना. इट्स इकोनॉमिक कॉस्ट है, इट्स एनर्जी कॉस्ट ऑन द सेल उन्हें स्टोर रखना अगर डीएनए रेप्लिकेशन अब अगले 36 या 48 घंटे के बाद होनी है. डिड आई आंसर योर क्वेश्चन?
यस सर. एक छोटा सा क्वेश्चन और था कि while we are doing the proofreading, you mentioned कि हमारे जो adjacent bases होती हैं, like for example अगर कोई mismatch है तो वो remove होती हैं and then they're fixed. So I have to ask कि like ये excess जो हमारी bases होती हैं, like are these part of the DNA polymerase, और जो हमारी mismatched होती हैं उनके साथ होता क्या? Like I don't know वो जाती किधर है, like वो bases.
So they are removed. So for example, here, oh, sorry. So here you have a mismatch. Right. So here you have a mismatch. DNA polymerase with its proofreading enzyme came in and it removed this one. This particular nucleotide is thrown out, discard कर दी जाती, इसे bin में फेंक दिया जाता है, and replaced with the right one. Clear?
Yeah, I was just concerned ke ye, like, do we have an infinite supply of nucleotides coming in from the DNA polymerase?
So nucleotides are not provided by DNA polymerase. DNA polymerase is an enzyme which is going to bring in nucleotide. Nucleotides हमारे न्यूक्लियस में प्रोवाइड की जाती हैं जब हम डीएनए रेप्लिकेशन हमने करनी है. तो लेट्स से वी आर गोइंग टू एम्पलीफाई, वी हैव टू कॉपी 6 बिलियन बेस पेयर्स. ठीक है, अ डिप्लॉइड सेल जो है वो 6 बिलियन है. 6 बिलियन का मतलब है कि 6 बिलियन हमने ऐड करनी है कॉम्प्लीमेंट्री. तो 6 बिलियन तो मिनिमम हमें चाहिए होंगी, but they are not limiting. They are always in DNA replication stage पे histone proteins, क्योंकि वो nucleosomes unwrap हो रहे हैं, new nucleosomes आने. They are enormous. Nucleotide synthesis routine में भी हमारी बॉडी में हो रही होती. Did I answer your question?
Yes, sir. Thank you. Makes sense.
Any other questions? Any other question? So if there are no more questions, I look forward to see you tomorrow. I hope Abdullah feels better.